Good day, everybody. We're continuing with this section on fungal diseases on plants and various examples. Today we're looking at Plasmodiophora brassicae. Plasmodiophora brassicae is the causal agent of clubroot on various brassica hosts. In this photo, we see some of the symptoms on brassica hosts. In the center, we have the Plasmodiophora that actually forms plasmodia inside the roots of brassica crops. And if we work clockwise from the left corner down round, we see severe wilting and yellowing of a brassica crop. And then we see the clubbing of the roots of the brassica. So in the top right and bottom left, you can see that these roots are not normal roots. They've turned into what we call club roots. They are enlarged and they are not able to take up water and nutrients properly. So as with any other plant disease, we need to understand the factors that result in development of an epidemic or development of disease. And we're going to look at the pathogen, the environment, and the plant aspects. Let's start by looking at the pathogen. Plasmodiophora brassicae is an obligate biotroph. That means it cannot grow and reproduce outside of a living host. It is a plasmodiophromycete in the eukaryote supergroup Rhizaria. So it's not a true fungus, it's actually a plasmodiophorid, but we will discuss it under the fungi for the purposes of this lecture. Other important plasmodiophromycetes include Spongospora subterranea, which is the cause of powdery scab on potato, and you will deal with this in your third year, Polymyxa graminis and Polymyxa betae, and these are both vectors. Besides being plant pathogens, they are also vectors of important plant viruses. Looking at the life cycle of Plasmodiophora brassicae, let's start with primary inoculum. So the inoculum in the soil is the primary inoculum, and the resting spores are the propagules of primary inoculum. And these are released from clubbed roots, which start disintegrating, and then they subsequently release these resting spores into the soil. So as I've mentioned, the source of inoculum, one of the main sources, is contaminated soil, and these resting spores, as you can see in that electron micrograph, can survive in the soil for approximately 20 years. Now, contaminated soil can be spread from one field to another through machinery or implements, nursery material. So if the soil or the growing medium in the nursery is contaminated, then the seedlings that are planted out in the field are already contaminated or infected with the pathogen. Wind-blown soil is another way in which the contaminated soil can be spread from one field to another, and waterborne spores flowing or running off from one field to another. Additionally, spores that are attached to the surface of propagative material, such as seeds, or in the case of Spongospora, tubers, also serve as inoculum sources. And the last one is contaminated manure. When animals are fed contaminated or infected plant material, the pathogen goes unhindered through the digestive tract and then comes out on the other end as manure that is contaminated with the pathogen. So, if we have a look at mitigation methods or measures for Plasmodiophora brassicae, we're going to start by looking at how do we reduce inoculum if it's in contaminated soil. So we can test soil. We can test it by means of a conventional PCR or a qPCR. We can also use bait plants to give us an indication of whether the pathogen is actually present and alive or viable in that soil. For machinery or implements, it's very critical that they are cleaned and sanitized so that we do not spread the pathogen from one field to another. When purchasing nursery material, the grower should check the supplier and ensure that the supplier, the source of nursery stock, is from a trusted supplier with no source of contamination. Wind-blown soil and waterborne spores are a little bit more difficult to control, but in this sense, it's important to limit erosion so that the pathogen is not spread by wind or by water. Then in terms of seeds that perhaps have spores attached to them or present on them, commercial seed cleaning can be used, treatment of seed with products that contain fungicides which are effective against Plasmodioptera, or some ways in which this source of inoculum can be reduced. And then regarding contaminated manure, if one is purchasing manure, the source of the manure needs to be confirmed free from the pathogen. And if producing your own manure, one needs to ensure that you do not feed infected material to livestock. If we're dealing with nurseries, how do we actually ensure that nurseries stay free from the pathogen? Well, one can use high pressure washers to clean seedling trays or equipment and then steam sterilize or disinfect them to reduce the inoculum load and avoid the buildup of organic matter in the disinfectant solutions. So it's basically about cleaning to get off all the organic matter and then applying a disinfectant solution. So the disinfectant should be replaced regularly because any disinfectant is deactivated by organic matter. So an example that could be used here is sodium hypochlorite, can be applied to the wash water or the sprays that are used to clean the nursery trays. One should avoid the use of water from dams for irrigation of nursery stock because often the pathogen is actually found in the water that is in the dams. However, if one needs to irrigate from dams, the irrigation pipe should be placed into the stillest part of the dam and the water should be collected from the surface of the dam. And this is because the resting spores are relatively dense and they sink to the bottom of the dam if the water is not disturbed. It's also very important when running a nursery to ensure very restricted access and ensure that there are vehicle or other machinery washdown facilities before entering and leaving the nursery. Again, the run of water needs to be managed and treated properly so that it does not flow back onto the farm. So coming back to the life cycle of Plasmodioptera, we now know where the primary inoculum comes from, but what happens once a plant is in the field? So the resting spores will start germinating in the presence of root exudates. And when they germinate, they produce zoospores. The zoospores then go on to infect root hairs, and this is the primary inoculum phase of this disease cycle. This process from germination, in other words, from establishing that there are root exudates and chemotaxis happening, so germination of resting spores through to infection of root hairs and formation of a plasmodium only takes about 4 days. After that, the plasmodium starts spreading in the root hairs and further into the root, and it enlarges. This takes about 3 days, so it's approximately a week from when the plant exudes root exudates and the pathogen it triggers the pathogen's response to when infection has occurred and there's some establishment of the pathogen in the host. After another 3 days, zoospores are produced and they are then ready to be released from the plant again. So within a 10-day period, we have already got infection, colonization, and the beginning of production of secondary inoculum. So it's very short. Right, so resting spore infection is triggered by temperature, moisture, pH, root exudates both from hosts and non-hosts, and then the calcium and boron concentrations in the soil. This infection of the root hairs is a prerequisite for secondary infection. So if primary infection of root hairs, in other words, doesn't take place, we do not find infection or secondary infection taking place. So the infection of root hairs occurs in hosts and in non-hosts, and it occurs in both resistant and susceptible hosts. So this can occur in basically any plant. this primary infection. But obviously in non-host, this is where it's going to end, as well as in resistant or tolerant hosts. It will not develop any further than production of a plasmodium. The secondary infection cycle is what is very important in terms of the disease and the reduction of yield and symptoms on the plant. And this occurs in susceptible hosts. So this is where the clubs on the roots are actually formed, where the zoospores are released through pores, they form a zygote, the zygote infects the roots, and then continues to form a secondary plasmodium. This plasmodium becomes multinucleate and expands and then fills the cells of the roots, turning them eventually into clubs. So the difference between what happens in a susceptible host and in a resistant host or a non-host comes in whether there's host resistance or not. So host resistance is obviously the most important management tool for Clubroot, and the resistance of the host is usually race or pathogen pathotype specific, which means it's a gene for gene interaction, and the resistance is quite easily eroded or overcome by the pathogen. So it's an arms race. Man has to continually develop and breed new brassica varieties that are resistant to Plasmodiophora brassicae. In this case, resistance stewardship, in other words, ensuring that resistance doesn't break down quickly, is very important. And to do this, it's critically important to know which pathotypes are present in a specific area. So research on this pathogen is important. So we need to know, for example, which pathotypes are present in South Africa. So I said infection of root hairs is also dependent on temperature. The minimum temperature for infection of root hairs is 10, the maximum 30, and the optimum temperature quite warm, between 23 and 26. So this is normally a disease that occurs in summer. And clubroot severity can decline as the initial infection of seedlings is delayed. So in other words, the longer the pathogen takes to infect the seedlings, the older the plants get, the lower the final amount of clubroot. So we want to try and delay initial infection to reduce the final amount of disease. And this can be done by changing planting dates to either plant earlier, in other words, earlier towards the end of winter, beginning of spring, or much later when it's in the middle of the rainy season and temperatures are cooling down already. And this is to avoid periods of high soil temperatures that are favorable for the development of, or for the infection of root hairs. Soil moisture and bulk density are also important in terms of movement of the pathogen through the soil and infection of root hairs. And it has been shown that clubroot severity is positively correlated to soil bulk density and micropore size. So growers should avoid planting in areas with compacted soil or waterlogged soil. High soil moisture is favorable for clubroot development. However, if saturated conditions occur for an extended period of time, this can actually reduce clubroot development due to conditions of hypoxia, in other words, lack of oxygen. pH plays an important role in the development of clubroot, and clubroot is normally most severe in acidic soils. So in other words, where the pH is less than 7. So growers generally want to maintain a pH above 7.2 by liming, for example. Research has also shown that the application of calcium and boron to increase the pH reduces infection. So at low pH levels, in other words acidic pH, the disease will be quite severe. As the pH increases, the disease severity comes down, irrespective of the calcium application. At intermediate pH levels, so around about 7, there is definitely a role for calcium in management of the disease. So basically what this says is calcium plays a role in management of the disease, but not at higher pHs, only at intermediate pHs and at relatively low pHs. This also depends on the soil's buffering capacity, the organic matter compound percentage, and the calcium-magnesium ratio. However, even if the soil is alkaline, it will not prevent clubroot development if there's a very high initial inoculum and the environmental conditions are favorable for disease development. So when looking back at the life cycle of Plasmodiaphora brassicae, we need to identify the weakest link. Where is it that we need to target this pathogen in order to result in disease control? So the zoospores are the weakest link because they do not have strong cell walls, they do not have multi-layered cell walls. So if we can target the zoospores, and particularly the secondary infection production of zoospores, we can lower the amount of final disease. So how is this done? Well, there are a number of synthetic soil treatments that can be used. However, none of them have been shown to be 100% effective in managing the disease. In addition, soil treatments by fungicides are quite particularly affected by soil type and the pathogen population. If there's a very high pathogen population, even a synthetic fungicide is not going to manage the disease. But some of the chemicals that are used and that have shown to be effective, although not 100%, are cyazofimid, phenamidone, mancozeb, fluazinam, which we need to bear in mind is phytotoxic and is actually being removed from the market in many countries. Flusulfamide, hymixazole and prosamidone, PCNB, metamsodium, and calcium cyanamide. Fluazinam and flusulfamide are actually only effective if you have a weakly virulent pathogen population. If you have a highly aggressive pathogen population and a high inoculum load, these two fungicides will not be effective. The other option that has been considered and been shown to be quite effective in the field is the use of biological control agents, or BCAs. And these BCAs target the peak releases of zoospores. So there are certain biocontrol agents that have shown potential in the past and that are actually currently being used in various countries throughout the world, including South Africa, to manage clubroot on brassica crops. The biological control agents work in a number of ways. They colonize root hairs, so they provide direct competition for plant surface to the pathogen. So the pathogen actually has to compete for an infection spot on the root hairs. The BCAs become endophytic in the plant, and in so doing, they induce resistance, the plant's resistance, through upregulation of various pathways and genes. And these include the jasmonic acid pathway, salicylic acid, and ethylene pathways. What's also important to bear in mind when dealing with Plasmodiaphora brassicae is that the rotation cycle plays a role. It's important to remove volunteer plants and weeds, particularly brassicas, as these serve as sources of inoculum between host crops. It's advisable to only plant hosts once every 4 to 6 years. This is not always possible, but this would be the ideal. Or to plant with non-hosts or leave fallow for 1 to 3 years to reduce the resting spore concentrations in the soil. So where possible, non-hosts are normally planted. The use of bait crops has been suggested in some studies, but it is unlikely to have a really significant effect on resting spore concentrations in the soil. So if we put this all together and we have a look at how we can actually manage plants to provide us with healthy crops, we can have a look at long rotations, various soil treatments, good soil moisture management, hygiene and sanitation, resistant cultivars, correct nutrition of the crop, and the correct rotation crops.